The small intestine is the main site of chemical digestion by enzymes and also the main site of absorption of nutrients and water. It is approximately 10 feet in length and 1 inch in diameter. It is located between the stomach and the large intestine, so that chyme from the stomach must travel the entire length of the small intestine before arriving at the large intestine. The small intestine consists of 3 regions: the duodenum, the jejunum, and the ileum. Figure 23.18 highlights the regions of the small intestine, beginning at the duodenum, which receives chyme from the stomach. The chyme then travels through the jejunum and then to the ileum, and at the end of the ileum, the chyme is delivered to the large intestine. The small intestine receives digestive juices and enzymes from both the liver and the pancreas. The bile duct from the liver and the pancreatic duct from the pancreas connect to the duodenum of the small intestine. A small smooth muscle sphincter called the hepatopancreatic sphincter regulates flow of bile from the liver and pancreatic juice from the pancreas into the duodenum. Typically, these are secreted into the duodenum during and after we have consumed a meal. The ileum, which is the distalmost region of the small intestine, empties into the cecum of the large intestine. A small valve called the ileocecal valve controls the flow of food residue into the cecum. The mucosa of the small intestine has elaborate circular folds along its length, which forces chyme to move in a spiral-like motion as it travels. This increases the time that food residue spends traveling through the small intestine. In addition, the circular folds also increase the mucosa surface area for absorption of digested nutrients and water. Figure 23.19 examines the circular folds surrounding the lumen of the small intestine, with part B of the figure in the lower left showing a microscopic sample of the folds. Additionally, the circular folds contain structures called villi, which are small projections of the circular folds that function to further increase its surface area. The mucosa epithelial cells, which are a simple columnar epithelium, also possess structures called microvilli, which are microscopic, finger-like extensions of the plasma membrane of the mucosa cells. The microvilli are often referred to as the brush border, as they give the cell surface a bristle-like appearance. On the small intestine mucosa, there are approximately 200 million microvilli per square millimeter, which gives the mucosa a massive surface area for absorption. In general, the small intestine produces 1 to 2 liters of alkaline intestinal juice, which functionally helps to neutralize stomach acids in the chyme. The enzymes operating within the small intestine work best at a higher or an alkaline pH. The chyme takes between 3 and 5 hours to be completely emptied from the small intestine into the large intestine. Returning to figure 23.19, we can see the villi of the circular folds and the microvilli on the surface of the mucosa cells. Part D of the diagram in the lower right shows a microscopic image of a few of the microvilli on just a small part of one of the mucosa cells. Once chyme has reached the end of the ileum of the small intestine, a small valve called the ileocecal valve, which is not shown in the diagram here, regulates the movement of chyme from the ileum into the pocket-like cecum of the large intestine. From there, chyme will slowly move through the length of the large intestine.